Edvotek instructional video, Understanding Quantitative Polymerase Chain Reaction. In this video, we're going to explore the fascinating world of molecular biology and explore a powerful technique called qPCR, which is short for Quantitative Polymerase Chain Reaction. Don't worry, we'll break it down step by step. First, let's talk about DNA, the blueprint of life. This important molecule contains the instructions that tell our cells how to function, but sometimes scientists need to study specific pieces of DNA to understand what's happening in a cell. That's where PCR comes in. So what exactly is PCR? Well, PCR stands for Polymerase Chain Reaction, which is a biotechnology technique that allows scientists to make millions of copies of a specific DNA segment. It is not a perfect analogy, but we can think of it like a photocopying machine for DNA. With qPCR, we take it a step further. Imagine you want to measure how many bacteria are in a water sample. qPCR doesn't just make copies of DNA, it measures how many copies are made in real time. This helps scientists figure out the level of bacteria that are present in a body of water. Here's what we need to perform qPCR. First, we need some DNA or RNA that was isolated from the samples we want to study. We add short synthetic pieces of DNA called primers. They are designed to be complementary to the specific part of the DNA we want to copy. Next, we need a molecule that produces a measurable fluorescent signal in the presence of our target DNA. This can be a dye, like cybergreen, or a probe, like TACMAN. As the DNA is copied, the fluorescent signal is detected. The more copies of DNA, the stronger the signal. We also need a thermostable enzyme called Taq-DNA polymerase. This enzyme builds new copies of the targeted DNA using the starting material as a template. We need free nucleotides. These are the A's, T's, C's, and G's that are the building blocks of DNA. Finally, we need a specialized machine called a quantitative PCR machine. Like a traditional thermocycler, the qPCR machine automates the changes in temperature. However, it also measures the fluorescence generated by the reaction. Here's how it works. First, all the components are combined into small, optically clear PCR tubes. The tubes are placed into the thermocycler. The mixture is then heated and cooled in cycles. The heat separates the DNA strands, and the cooling allows the primers to bind to their target sequences. After the primers bind, Taq builds the new DNA using the sample DNA as a template. At this time, the samples are excited using light at a specific wavelength, and the emission is measured. By measuring the strength of that signal, scientists can determine how much of the original DNA was present in the sample. This is useful for identifying bacterial contamination in water, studying gene expression, and even detecting pathogens like COVID-19. And there you have it. qPCR is an incredible tool that allows us to peek into the microscopic world of genes and molecules. Who knows, maybe one day you'll use qPCR in your own research. We hope this helps you achieve success in your next experiment. Thanks for listening. We pride ourselves on providing the highest quality customer service for our teachers. Call, email, or send us a message on social media.